Okay. Good
Is
evening,
it?
Mrs. Sweden, Mr. Tegar, and good morning, Professor Miguel. My name is Marcella Jessica from Universitas Gadjah Mada, and today I would like to present my thesis with the title Metadata Analysis and Systematic Review of Phytochemical Composition and Factors Affecting the Levels for Microalgae. I will be reporting the result through this presentation with the agenda consisting of the introduction of the study, the objectives, the method used, the result analysis, and the conclusion. First, I would like to explain about the background of the study. Indonesia is the fourth highest populated country in the world, resulting in productive land around 0.09 hectares per person. Along with population growth over time, land availability is decreasing for farming to meet food needs. Therefore, Indonesia still needs to improve their food security to fulfill the food consumption for the population. Food security is the availability of food in storage and markets. The potential solution for the food security is microalgae. Because Indonesia is an archipelago, therefore there are numerous oceans as microalgae source. Microalgae can be mass-produced easily in Indonesia. So, creating database about microalgae species in Indonesia and also their chemical composition is important as an information to develop food from microalgae. Data about factors affecting the chemical levels of microalgae, such as phylogeny and also the growth condition, also important to help in producing microalgae as food optimally. Compiling all of those data will result in a big data set, and it needs to be processed statistically. So, chemometrics method is used to work on the data and to process the variables to bring out the results needed to evaluate the data. Hence, the objective of the research are: first, to compile a comprehensive database on microalgae in Indonesia, including the varieties and the source of origins. Second, to conclude the chemical compounds profile of microalgae in Indonesia. Third, to determine which growth factors influence the chemical levels of microalgae in Indonesia. Fourth, to resolve how different microalgae relate to each other based on their major compounds. Fifth, to decide which major compounds distinguish the microalgae that are responsible for the majority of the variations. And last, to observe the use of major compounds for fingerprinting a specific variety of microalgae within the same species. To achieve all of these objectives, several methods were applied. The first method was for data collection, using systematic literature review with the SALSAR framework. The first step is to define research protocol, to define the study scope, which is the chemical composition of microalgae in Indonesia. The second step is to select search strategy. The keyword used were species obtained from the LIPI website, the word Indonesia to make sure that the sample was obtained in Indonesia, and also the chemical compounds, such as major, pigment, and the database search were Google Scholar and Scopus, and approximately 11,000 papers appear in the search results. The third step was appraisal. First, the studies were selected using this inclusion and exclusion criteria, resulting in approximately 400 papers attained. And then, the quality of papers were also assessed using abstract and main body scheme reading, so a total of 211 final papers were kept. The next step was synthesis. The data was extracted and exported to the Excel sheets using this category. And then the data was analyzed using chemometrics method, and the final step was report writing. Next, I will explain the data analysis using the chemometrics method. Before the data analysis, the unit was standardized, which compared to all of the units was percentage and on dry matter basis. The first chemometric method used was hierarchical cluster analysis. It was done to 113 microalgae samples using 5 variables of chemical compounds, which was carbohydrate, protein, lipid, moisture, and ash. The data was standardized, and the clustering method used average linkage and also squared Euclidean distance. The complete cases obtained was 19, which meant the data process was samples with no missing value on any of the variables. The second method was principal component analysis. It was done to the same cases and the same variables, and the data was standardized. The data was extracted by 3 components, and the missing value treatment using listwise, resulting in 19 complete cases. The third method was spiroweb analysis. It was done to 5 samples of Spirulina potensis using the same 5 variables. Both actual scale and standardized scale were used. All of this method was done using stat graphics application, and the following results were obtained. First, the systematic literature review resulted in database consisting of 71 microalgae species in Indonesia, mostly come from Chlorophyta phylum, followed by Heteroconophyta and Cyanophyta. Also, most of the samples were collection from 2 institutes in Java, and around 85 papers did not provide any information on the sample's origin. Meanwhile, the chemical composition, around 865 chemical values were obtained. The data cover 46% of major compounds, 35% on trace, and 19% on pigment. It was reported that in microalgae, major compounds consist of carbohydrate, lipid, protein, and others. Trace compounds in microalgae, polyphenol, phytosterol, vitamins, minerals, and also others. Also, the pigment content were chlorophyll, carotenoid, and phycobilprotein. 71 species, major compounds was in 60 species, and trace and pigment in 29 species. However, only 6 species have comprehensive data on their chemical composition. They are Chlorella, Chlorella vulgaris, Donaliella salina, Nanochloropsis, Spirulina, and Spirulina potensis. Some of them has generally recognized as safe status, approved by the FDA, and also some are categorized as no toxins known. Their recognized status might be the reason why they were more explored than the other species. In addition, Chlorella, Donaliella, and Spirulina are microalgae that dominate the market, and Nanochloropsis also has commercial importance. These are the major chemical compounds that were reported in each species. Chemical compounds in microalgae also can vary, and it depends on multiple factors, such as the growth factors. 71 papers examined the effect of growth conditions on 8 chemical compounds of major and pigment, and it was found that there are 6 growth factors that affect the microalgae chemical content. The first is harvest period, whether microalgae is taken in exponential or stationary phase. The second is light factor, whether the intensity or the lamp type or the photoperiod. The third is the nutrient of the medium, both major and minor. The fourth is the pH of the medium, whether acidic, neutral, or alkaline. Fifth is the salinity of the medium, and last is temperature, whether high or low. Next, I will explain the result of the chemometrics method. The first method was cluster analysis to examine how different microalgae relate to each other based on their major compounds. The results showed two big clusters created by limiting linkage distance at 6. The first cluster consists of chlorophyta and heteroconophyta. The second cluster consists of cyanophyta, and there are three remaining species that each come from the three phyla, but by looking at their linkage distance, they are very far connected to the first two clusters. By examining further, it was found that this formation of clusters were more influenced by phylogeny rather than their growth conditions. The first cluster is eukaryotic microalgae, and the second cluster is prokaryotic algae. It was also found that same genus and species tend to group in advance, although cultured in different growth medium, such as the Spirulina plantensis number 4 and 6. Also, they ended up joining in the same cluster, although being separated at first, such as with this Spirulina plantensis number 11. The next method was principal component analysis to determine which major compounds distinguish microalgae by looking at the majority of the variation. The PCR result represented 97% of total variance, PC1 for 65%, PC2 for 24%, and PC3 for 6%. The PCR result showed agreed with the cluster analysis. There were two big groups of eukaryotic microalgae and prokaryotic microalgae, also three separate species. The first principal component separate the eukaryotic and prokaryotic microalgae by their ash and protein content, shown by the blue group and the red group. The second principal component separates species in prokaryotic by their protein and moisture content. This Spirulina plantensis group was marked by red, and another Spirulina plantensis was separated by its moisture content marked by orange. The third principal component separates the eukaryotic microalgae by their ash, lipid, and carbohydrate content. This blue group consists of chlorophyta and heteroconophyta, and Nanochloropsis number 16 was differentiated by its lipid content. It is a member of heteroconophyta, and Crucigenia quadrata is separated by carbohydrate. It is a member of chlorophyta. The third method used was spiderweb analysis to see the major compounds profile of 5 Spirulina plantensis samples. The spiderweb showed that indeed the major compounds profile of the same species can vary, but it is not clearly shown in the actual scale, except for Spirulina plantensis number 3 that is very high in moisture because it is the only fresh sample. The 4 others were dried. So, the standardized scale was used to see more clearly the difference in each major compounds. The scale used was the lowest value to the highest value of each compound. And it was found that chemical content in the same species varies because of different growth environments. Number 3 and number 4 was obtained in succohydro but differ in the sample forms. Number 6 were cultured in xerox medium. Number 11 was cultured in fertilizer medium, and number 16 was obtained in Yogyakarta. So it can be concluded that same species with different forms and different growth conditions contain different level of major compounds. The fingerprint profile using actual scale is insufficient for authentication of Spirulina plantensis in Indonesia. So the standardized scale is more suggested. After examining all of the results, it can be concluded that first, data about microalgae in Indonesia are available, but not yet compiled. It still lack on the origin of samples, the growth condition, and also the analysis method used. Second, the data on chemical compounds profile of 71 species in Indonesia consists of major, pigment, and trace, available for 72% of reported species from LIPI, but is still lack on pigment and trace content. Third, there are 6 growth factors that have effect on microalgae chemical content, which are harvest period, light, nutrient, pH, salinity, and temperature. Fourth, microalgae classification based on major compounds level correlate more to their phylogeny rather than the growth condition. Eukaryotic microalgae cluster separately from the prokaryotic microalgae. Fifth, the major compounds level can differentiate microalgae. Eukaryotic are differentiated by their ash content and prokaryotic by their protein content. And last, the same microalgae can vary in their major compounds profile because of different sample forms and different growth environments. And that's the end of my presentation. Thank you very much.